Here all sit straight. So, from enzymes, what did we learn in the morning? We have learned that all the enzymes are proteinaceous in nature. There are few exceptions. Can you name them? Ribozyme and ribonuclease protein. Ribonuclease protein. Those are extracted from RNA, ribonucleic acid. So, few of the proteins are of exceptions from RNA, of which ribozyme will help in peptidyl transferase enzyme, that will help in the formation of peptide bonds and viral replication as well. Ribonuclease P will help in cleaving or trimming of the 5' end of tRNA. And then we have learned the differences between organic catalyst and inorganic catalyst, right? Can you name a few, like 2 or 3? Catalysts, enzymes, specificity, sensitive to temperature. Okay, there which catalysts are more specific? Biocatalysts are more specific. They are all of organic compounds or organic compounds. And then temperature sensitive. Optimum temperature is around 40 degrees Celsius. That we have learned. And those enzymes which are extracted from thermophilic organisms, their active site or their activity exceeds around 80-90 degrees Celsius also, right? And then we have learnt on mechanism of enzyme action. Enzyme will have active sites. What are the active sites? Nothing but most of the enzymes are of what structured proteins? Tertiary structured proteins. When it is folded like this, some minute crevices and pockets will be there. That crevice to which into substrate is fit is known as what? Active site. And when substrate is binded to that active site, it forms what? Enzyme substrate complex. Enzyme substrate complex turning to enzyme product complex, that state is known as what? Transition state. And what energy is required? Activation energy. And at the end, EP complex will release out the products, right? Two mechanism of actions we have learned. What are they? Lock and key model, induced fit hypothesis. Which is more acceptable? Induced fit hypothesis, because of its flexibility, right? This is proposed by lock and key hypothesis, proposed by Emil Fischer. Induced fit model, Daniel Koshland. This is about mechanism of enzyme action. And then we have learnt on exothermic endothermic reactions based on the graph. In that graph, what is plotted on the x-axis? Progress of the reaction. What is plotted on the y-axis? Potential energy. If substrate is at high PE, product is at low PE, then what type of reaction it is? Exothermic reaction. If substrate is at low PE, product is at high PE, it is endothermic reaction. Got it, all of you? And then we have learnt on factors affecting enzyme activity. We have learnt on turnover number morning. What are the factors affecting? pH, temperature, substrate concentration. Where do you see bell graphs, bell shaped graphs? pH and temperature. pH is optimum for every other enzyme, but for all the organic catalysts, optimum temperature is around 40 degrees Celsius. Remember that. And after this, we have learnt on substrate concentration. You will get an inverted L-shaped graph, right, wherein you have learned something on Km, which is Michaelis-Menten constant, at which half of the Vmax is achieved. So, until reaching up to the saturation point, if you increase the substrate concentration, the rate of the reaction will be increased. Once it has reached the optimum, it will be stable or constant, right? Easy all? Yeah. Now, inhibition of enzyme activity. That's it, right? We have revised everything. Now, we have to learn on the inhibition, on cofactors, and on the classification of enzymes. Listen here. First is competitive inhibition. Competitive inhibition is nothing but, here inhibitor structure and substrate structure will be same. Inhibitor structure will be similar to what? Substrate structure. When it has a structural similarity with the substrate, it can easily fit into the active site. So, the competition is in between what? Inhibitor and substrate. Okay. In non-competitive inhibition, we have learnt there are many crevices, right, apart from active site. So, inhibitor will come and bind to one of the crevices, which will change the shape shape of the active site. Previously it is like this, now it is like this, so that it will not allow the substrate to bind with it. Here also it is competing with the substrate, but this is not directly competing with the substrate. Okay, inhibitor is binding to non-active site of the enzyme, which is changing the shape of the active site, which is not allowing the substrate to come and bind it to it. Right, this is non-competitive inhibition. Next is feedback inhibition or allosteric inhibition, which is nothing but, for example, glycolysis is there. What is the product of glycolysis? Pyruvic acid. If you already have abundant pyruvic acid, will it allow the initial step of the reaction? No. So the product of any reaction, if it is inhibiting the first step of the reaction, that is known as what? Allosteric or feedback inhibition. So now you got the basic meanings of all the types of inhibitions, right? Now let us move on to a comparative analysis. So first is about what? Binding site. Inhibitor binds to what site? Active site here. Here it binds to the site other than the active site. This is regulatory or allosteric inhibition, which is not the active site. Product is inhibiting the first step of the reaction, right? And then structural similarity. Inhibitor is same like that of substrate. Here inhibitor is not structurally similar to the substrate. Product or inhibitor, see, the ultimate product is nothing but the inhibitor. This may or may not resemble the substrate. So in these two types, substrate similarity not there. Here you will see the structural similarity with the substrate. Clear all? And then effect on Vmax, which is maximum velocity. We have seen this, right? This is what? Maximum velocity. Okay? It is unchanged. Maximum velocity of the competitive inhibition, when inhibitor is binded to, Vmax is what? Unchanged. But here if you see effect on substrate concentration, I have 10 molecules of the enzymes, 10 enzymes I have. Okay, I have 5 inhibitors, 5 substrates, both of them are competing. Okay, if I raise the substrate concentration to 6, more substrates will bind to enzyme, right? To 7, to 8. So to reduce the effect of inhibitor, we have to increase what? Substrate concentration. So what is the effect on Vmax in competitive inhibition? It is unchanged. The effect on substrate concentration is increased, right? Here the effect on Vmax will be decreased. Velocity of the reactions get decreased because it is affecting the active site of the enzyme. It is not allowing the substrate to bind. And substrate concentration is unchanged. There is no competition with substrate concentration. No, it has 5, 6 or 7 also, it will make no change because the active site of the enzyme is altered. This too you have to remember, very important. Effect on Vmax and effect on substrate concentration. Nothing to mug up here. There is a simple logic. Here in competitive inhibition, inhibitor is competing with substrate. So substrate concentration is increased. This will remain unchanged. But here if you see, Vmax is decreased because it is not allowing substrate to bind, and substrate concentration Km will remain unchanged, right? And here Vmax, it can increase or decrease. It is modulator. It is variable because it depends on what concentration? Product concentration. Here if you see effect on Km, Km is also variable, right all? Next, can be reversed by the substrate. Yes, competitive inhibition can be reversed by the substrate. If you go on raising the substrate concentration, enzyme will always obviously bind with what? Substrate. So it can be reversed, but this cannot be reversed. Non-competitive inhibition cannot be reversed. It is irreversible because it is affecting on what? Active site on the enzyme. This is not easily reversible, but this is regulated. Wherever you find the term regulatory, it has both the ends of the coin. Both the possibilities are there. Regulatory, it can have the positive effect, it can have the negative effect. Okay, so this is what? Regulatory. irreversible, reversible. Clear now? Is there anything to memorize more or mug up the things? No, right? This is the simple logic. If you understand the definitions, then all other things also you can remember easily. Okay? Now, the most important aspect is examples. For remembering this example, you have to once recall TCA cycle. Succinate dehydrogenase, where do you see in Krebs cycle, if you remember? Succinic acid to fumaric acid. Very good. So, succinate to fumarate. This enzyme is required. What is that? Succinate dehydrogenase. This substrate, what is this? Succinate, right? Inhibited by what? Malate. Malonate will inhibit succinate. Okay, so both of them are having what? Structural similarity. Malonate is resembling succinate. Okay, and to remember this example, you have to remember ETS, electron transport system. You have 4 complexes, right? Complex 1, if you remember, NADH dehydrogenase. Complex 2, succinate dehydrogenase. Complex 3, cytochrome BC complex. Complex 4, cytochrome C oxidase. So, this cytochrome oxidase is inhibited by what? Cyanide, whose effect is irreversible. Right? TCA cycle, one example we have taken. What is the enzyme? Succinate dehydrogenase. Inhibitor is what? Malonate. It shows a structural similarity with succinate. Here if you observe, cyanide will inhibit cytochrome oxidase. This effect is irreversible. Got it? Next. Here if you see, this is from glycolysis. ATP inhibits phosphofructokinase, isoleucine inhibits threonine deaminase. This is from amino acid synthesis. What is inhibiting? Isoleucine. ATP, I will tell you. Third step of glycolysis. Fructose 6-phosphate to fructose 1,6 bisphosphate. That will give out what? ATP energy is required. If more amount of ATP is already present, will it allow this reaction to happen? No. So, ATP is inhibiting what enzyme? Phosphofructokinase. Got it? Which is third step. This is also known as rate limiting step in glycolysis. When we are discussing that, I will tell you detailedly why we are calling it as rate limiting step and all. As of now, what you have to remember? 3 examples. One example for competitive inhibition is malonate competes with succinate for the enzyme succinic dehydrogenase. This is from TCA cycle. Second is from ETS, non-competitive inhibition. Cyanide is inhibiting cytochrome C oxidase. Next, feedback inhibition. ATP inhibits what enzyme? Phosphofructokinase. Clear right till here? And then enzyme inhibitor complex is formed here. Here it can bind both to the free enzyme and it can form EI, or it can bind to enzyme substrate complex also. Because the whole point is what? Changing the active site of the enzyme. It can bind to enzyme or ES complex. Here also the complex is formed, but usually this induces what? Conformational change in the enzyme. Okay. Till here you remember, this is an extra information, it is up to you. Use in medicines. This is used in drugs, competitive inhibition, like producing sulfa drugs or PABA. And then this is used in toxins and drug actions. These are used to regulate the metabolic pathways. This is up to you. But till here though, you have to remember each and every point. Got it? Except remembering examples, there is nothing much to memorize from the inhibition part of the enzymes. Okay, clear all? Next. Yeah. Classification of enzymes. Here you see classification of the enzymes. First is oxidoreductases or dehydrogenases, first class. Before letting you know about this, International Union of Biochemistry, IUB, around 1962 like that, they have named the enzymes. with 4 digits, 1, 2, 3, 4, of which first digit is for class. If I have to take some enzymes like lactate dehydrogenase, its indication start with what? 1. Some enzyme related to transferase like hexokinase, it starts with what? 2. So first is remembering what? Class. Second is subclass, subclass, and fourth is the actual enzyme. What you have to remember? Should you remember all the subclasses, subclasses? Not required. Just you remember 4 digits. Each digit, 1 is for class, 2 subclass, 3 subclass, and 4th is directly of what? Enzyme name. Okay. So here if you observe, you remember only first 6 classes and few examples that we are learning here. You can easily figure it out. Okay. First class is what? Oxidoreductases or dehydrogenases. Here, if you look into this, reduced, oxidized. This gets oxidized and this gets reduced. Where redox reaction happens. In a single reaction, oxidation happens, reduction happens, like our photosynthesis reaction. Enzymes which catalyze oxidation reduction between two substrates is known as what? Oxidoreductase enzyme. That which catalyze these are what? Oxidoreductases or dehydrogenases. Example is lactate dehydrogenase. Second is what, ma? Transferases. Enzymes catalyze a transfer of group between a pair of substrates. For example, this substrate is binded with one group, let us say any functional group, and one more substrate is there. This group is transferred from this substrate to this substrate. That group can be anything, OH group it can be, hydrogen or proton, it can be a functional group, anything it might be. So transfer of any group from one substrate to other substrate. What enzymes are those? Transferases. Example is hexokinase. Next, hydrolases. Enzymes catalyzing hydrolysis of esters, either peptide, glycosidic, PN bonds, etc. All these are what? Hydrolases. Best example is peptidase. Second and fourth are what? Lyases. Enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis. Other than hydrolysis, if it is included in removal of any of the groups, that are what? Lyases. Example, aldolase. Next is what? Isomerases. Interconversion of any groups, which includes optical, geometric, positional isomers. Number of carbons will be there, same. All elements will be same, but positions will be shifted. Like glucose 6-phosphate, fructose 6-phosphate. Those are what? Example, triose phosphate isomerase. You can easily identify isomerases because at the end isomerase term will be there. Ligase. You can easily tell me what is ligase. Enzymes that catalyze linking together of two compounds. Example, pyruvate carboxylase. Remember, so these are the examples and 6 classes. First is oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. OTH, LIL, LIL. This is the abbreviation you can remember all the classes in order. Oxidoreductases, in order you can remember, right? So clear on the inhibition, clear on the classification, right? So here are few more examples. Oxidoreductases, oxidase, reductase, catalase, peroxidase. All this comes under the first class. This part, examples part, everybody have to remember from this slide. Transaminases, kinases, transferase, protease, amylase, lipase, all these are under hydrolases. Simple example to remember hydrolases. In cell the unit of life we have learnt about one organelle which is rich in hydrolytic enzyme, lysozymes. It is rich in what enzymes? Hydrolytic enzymes. Protease will cleave proteins, amylase carbohydrates, lipases lipids, maltase carbs, nuclease nucleic acids. Okay. Next, liases, aldolase, carbonic anhydrase, decarboxylase, this and all will come to picture. Isomerases, at the end you will have the suffix isomerase, easy to remember. Ligases, RUBP carboxylase, ligase, phosphoenol pyruvate. These are the examples. Okay. Now, so all the enzymes are proteinaceous in nature, we have discussed, but enzyme to perform its function actively, okay, it has two parts in the enzyme. Okay, enzyme has two parts, one is protein part and the other is non-protein part. Enzyme has two components in it. One component is protein part. Protein part of the enzyme is known as what? Very good, apoenzyme. Non-protein part is cofactor. This cofactor can be a prosthetic group, coenzyme or a metal ion. Okay, fine. So now non-protein constituents are called as what? Cofactors. Cofactors, they are bound to the enzyme to make the enzymes catalytically active. Three kinds of cofactors are identified: prosthetic group, coenzymes, metal ions. Protein portion of the enzyme is what? Apoenzyme. Just to remember this flow chart, it is enough. Oh, it's gone, it's okay. Here it is there. Just remember this, it is enough. Yeah. So the example of first prosthetic group, if you see how they are binded to the protein part. So this is apoenzyme, right? They are tightly bounded. Prosthetic groups are what? Tightly bounded. Example you see, peroxidase, catalase, they help in breakdown of what? Hydrogen peroxide to water and oxygen. Okay. What is the prosthetic group? Heme is the prosthetic group, that is a part of the active site of these enzymes. Okay. Firstly, remember the examples. Peroxidase, catalase, it has prosthetic group. What is the prosthetic group? Heme. It will help in catalyzing hydrogen peroxide to water and oxygen. Clear with the prosthetic group? Second, how prosthetic groups are binded to the apoenzyme? Tightly binded. Coenzymes. Coenzymes are not that tightly binded, they are transiently binded. Look at here. Coenzymes are also group of organic compounds. What are coenzymes? Organic compounds, but their association with apoenzyme is only transient. Usually occurs during course of catalysis. Only at the time of catalysis they are binded, and they are transiently binded, means it is temporary bonding. Okay. The essential chemical components of many coenzymes are what? Vitamins. They are transiently binded, and essential components are what? Vitamins. Coenzymes serve as cofactors in a number of different enzymes catalyzed reactions. Coenzymes, best example if I have to take, NAD, nicotinamide adenine dinucleotide, and NADP. Tell me full form of this. Nicotinamide adenine dinucleotide phosphate will have the coenzyme that is niacin. Prosthetic group for the previous enzyme, peroxidase and catalase is heme. Here it is what? Niacin. What you have to remember for coenzyme? These are organic compounds, transiently binded. Mostly they are vitamins. One best example is niacin, which is for what? NAD, NADP. They contain niacin. Metal ions. The number of enzymes require metal ions for their activity, which form coordination bonds. Prosthetic group is tightly binded. Coenzymes transient bond. Here metal ions are binded by what? Coordination bonds with side chains at active sites of the enzyme, at the same time on the other group, on the cofactor. Cofactor and here both have what? Coordination bonds. Best example is zinc is cofactor for carboxypeptidase enzyme. Peroxidase catalase, heme. NAD, NADP contain what? Niacin, that is for coenzyme. Metal ions, coordination bonds it is binded by a coordination bonds. Example, zinc to carboxypeptidase. Clear, no? Take 5 minutes time, revise a bit, scroll the chapter. We are going to start the PYQs. cellular constituents, secondary cellular constituents, secondary metabolites, inorganic elements, Shall we start with the objective? Yeah. First question to this row. We'll make 3 groups. 1, this 2 rows 1, 3 third group. Okay. You can refer and answer. Exactly 1 minute time for each group. The other group can take over if 1 minute is completed. It's right away. The answer is right away. Done? Group 2. Lysis. One substrate is to 2 products. Yeah. Second question. We have got almost of 4 questions in your previous NEET. Recently happened, NEET 2026. First option? Yeah. Trypsin? Enzyme. Morphin? Alkaloid. Concanvaline A? Lectin. Collagen, intercellular ground tissue. Only from proteins you got 2 questions. Alpha helix is found in which level of protein structure? Secondary structure. Alpha helix and beta sheet? Secondary structure. This question, group 1. Easy to answer, you can. So we need to identify correct statements regarding amino acids. They are substituted methanes, correct, right? Serine is an aromatic amino acid? No. What are the 3 aromatic amino acids we see? Phenylalanine, tryptophan, tyrosine. Valine is a neutral amino acid? Correct. Lysine? Basic. Tell me few basic amino acids. Lysine, arginine, histidine, valine. Valine neutral, right? Serine. Okay. So this is wrong. What are the right options? A and C. Got it? Next. Five questions. Single paper. Last question. Last question of today's session. Identify the correct statements about biomolecules. Lipids are water insoluble. Wrong. Whenever statement based questions are given, without hurry, first look at this properly. They are asking for correct or incorrect statements. Just if you are making here wrong, then an entire base goes wrong. So look at here properly and then start reading the options. Here he asked us to identify correct statements. Lipids are generally water soluble. Wrong. Proteins are polymers. True. If he is giving homopolymers, it is wrong again. Polysaccharides are long chains of sugars. Adenine and guanine are substituted pyrimidines. No, they are purines. Almost all enzymes are proteins. Correct. So the right answer is B, C, E. Okay. And in your NEET paper, when question paper, or here also whenever question papers are given, that question paper is for usage only. If this is given, encircle it. Correct statements. So, beside the statements, keep it whether right or wrong. That will help you to identify the option easily. You will just scroll with eyes as if you should not touch that paper. Don't do that next time. From now you start working on the paper, it will be easy for you. You will make less errors. Okay. So as of now we have solved around 6 questions from a single paper. I want you people to revise the entire chapter thoroughly. Tomorrow we'll practice from 2025 and re-NEET questions also I will get, and we'll practice it up to 2019. Okay. So come prepared. Without preparation, I felt like this will not be a good session. So come prepared tomorrow. We'll have a very nice PYQ session. Okay.